And so many of my colleagues, including ourselves, start to think that Alzheimer actually is a cardiovascular disease. It's something associated with what is in your blood rather than what is in your brain. In biology, it's not a simple additive process. It's not that 1 + 1 is always 2. 1 + 1 might become even more than 2 because there are sort of phenomena that characterize life in general, which is called emergence. Is this fact that by putting things together, you create a new type of energy that come from the collectiveness of things.
Thank you for joining Once Changing the World. I'm your host, Eddie Avil. And today, I'm delighted and honored to have with me Giuseppe Battaglia, ICREA professor who leads the Molecular Bionics Group at the Institute for Bioengineering of Catalonia. A pioneer in molecular bionics and nanomedicines, he investigates how soft matter self-assembles at the nanoscale to mimic and enhance biological processes. Battaglia uses molecular engineering and nanotechnology to redesign structures like viruses into versatile biotechnology platforms. His recent breakthrough, in collaboration with the West China Hospital of Sichuan University, demonstrated a nanotechnology approach that rapidly clears amyloid peptides, restoring brain vascular functions, and reversed Alzheimer's pathology in a mouse model. So, Professor, really appreciate you taking time and being part of the podcast. 57 million people globally suffer from Alzheimer's, and there's roughly around 8.8 million people here in India who suffer from Alzheimer's. So yes, obviously it's a huge problem, but not many clearly understand what Alzheimer's is. So will be great if you could start with maybe, making the audience understand what Alzheimer's is and what are the underlying causes of Alzheimer's.
Sure. Yes. Thank you very much for having me here. It's really nice to be in this podcast. Alzheimer is a disease that it was discovered at the beginning of the 20th century by looking at the brain of patients that were experiencing some really strange cognitive decline, etc. It's been, however, really studied in the last 20, 30 years more and more, and has become a clear priority in healthcare because as today it is the seventh cause, the seventh most common cause for death in the world, the second most common cause of death in the high income world, and is becoming more and more so with an incidence that is increasing considerably. You talk about 57 millions right now in the worldwide. By 2050, we're talking about prediction of 160 millions people will be affected by Alzheimer. It is a disease that is very long lasting. In some cases, it can go all the way for 20 years with consequences in the lifestyles of the patients and the people caring for the patients. So the cost associated with the disease is not just social and emotional, but economically, we're talking about in the order of trillions of dollars per year to care for people. The causes are still very much a subject of study. There are a lot of indications that certain factors associated with lifestyles have a huge and higher risk of Alzheimer and dementia, particularly cardiovascular factors such as high fat, smoking, and certain type of pollutions, etc., or abuse of drug, etc. There is a correlation with certain events, such as, for example, brain injury. There is a lot of correlation between those and some Alzheimer forms or even other forms of dementia. Clearly, something happening at the brain, and then all of a sudden the brain loses some of the functions. And unfortunately for us, this loss is not instantaneous. It's not like an acute type of response. It's very slow and happens in the order of 20 years. And by the time you realize a patient or individual is affected, it's already quite late in the stage, and sometimes it's very difficult to reverse that damage caused. Historically, Alzheimer was associated with certain protein accumulation in the brain. Now things are moving on, and there are new other possibility and new other hypotheses how Alzheimer's occurs and evolves clinically. Yeah.
Yes, Alzheimer's is such a big problem, globally, and you said it's growing, and it's just progressively going to grow. I mean, so little we know about the brain. I mean, the brain it's one of the most complex organ. But we know so very little, and I guess, I mean, neuroscience, neurotech, and researchers such as yourself are like constantly making progress to kind of understand the complexity of a brain and kind of figure out how we can kind of probe it, with neuroscience, neurotechnology to kind of reverse these kind of things. Before we get into how you manage to kind of reverse Alzheimer's in a mouse model, will be great if you could explain nanotechnology, because I think nanotechnology sounds very science fictiony. Because when somebody says nanotechnology, all we get to know is that you are kind of tinkering around with or you going into the nano world, which is like the really 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 tiny world. So for my listeners who don't really know what nanotechnology is, it'd be great if you could start with explaining what is nanotechnology and how is nanotechnology currently being harnessed or leveraged in medicine and healthcare.
So nanotechnology was born in the late 80s and the beginning of 90s out of the ability that we developed in the labs to see molecules with much higher precision, right? The different electron microscopes, and particularly atomic force microscopes, allow us not only to see molecules, but also to manipulate them. And so all of a sudden, a new way of creating material was proposed. A way that not only use chemistry as a power, like we do with many other things. So we just make things reacting and creating new molecules, new materials, but also the ability to control how these molecules assemble in space. What is the structure properties of these materials? And we've been very good at creating structure which are not particularly big. And this is why the word and the prefix nano is being used for defining this concept, because effectively allow us to engineering object down to molecule by molecules. And this ability allow us to create new, complete novel functions. And there are cases like, for example, the carbon nanotubes. By creating these sort of beautiful tubes or arrangement of carbon, you create materials that have a resistance which is stronger than anything else on Earth. Or quantum dots, a few years ago, was awarded a Nobel Prize, where by arranging molecules of semiconductors materials, you start to create photonic effect that creates lights and tunnel lights in a very unique way, allowing us to create even more powerful lasers and display, etc. And so it is very much an evolution of chemistry. Chemists still are involved in nanotechnology because you still need to know how to manipulate matter at the chemical level, but there is a lot of physics coming into play by looking how molecules are arranged within each other, right? And so it effectively becomes like a Lego brick games where you can really build materials and structure with a very high level of molecular precision. In medicine, this has created some very interesting properties. Don't get me wrong, nanotechnology is also what nature does already. Many of the structure in our body, including proteins, vesicles, and lipoproteins, and even viruses and bacteria and cells, are effectively a great example how arranging molecule in a certain space at the nanoscale, you can get a very unique function. So nanotechnology, so biology effectively has already been using nanotechnology approach for many years. So what we learn is that if we can copy that kind of arrangement of molecular space by using synthetic materials or perhaps materials which are hybrid, synthetic and biological in origin, then you start to have a new way to manipulate biological responses. And so this is basically what many people from the nanomaterials and nanotechnology world have tried to do for many years now, to really translate this new type of engineering into solve a problem associated with medicine. Of course, the three areas which are very critical, one is understanding and probing biology more cleverly, so creating new type of diagnosis, new type of way to read the biological output and to find out whether something is wrong or not, so to identify diseases or not. Then there is an area where is effectively you solve a critical problem, which is very important in medicine, how to get to the right place in the body, right? How to navigate the body. It's kind of inspired in many ways by sci-fi, such as the Fantastic Voyage, this idea of creating a device that, like a submarine, can really go into our bloodstream and get I don't know, to a tumor and only kill the tumors, or to the brain and only cure the brain, etc. This is very much what drives the things. And so there is a huge input into this type of material design in order to create a sort of carrier and create a much more efficient way to deliver drugs, but also to enable complete new therapy. Thanks to nanotechnology, we've been making huge progresses in genetic therapies. The ability to cure disease by manipulating and altering the genetic DNA, the nucleic acid of a cell, so the genetic materials of a biological system. And a good example of that was COVID vaccine. The way the vaccine went so quickly and created such a huge impact in the society was because we created some new nanomaterials able to deliver a new type of genetic cargo called mRNA, messenger RNA, that was able then to transform into the protein that is needed for the vaccination process. And that really had a huge impact. And now the genetic therapy is having an impact. More recently, and this is particularly what my research group is now focusing their attention on in the last 5 years or so, we now identify this completely new way to create function and biological function by creating sort of super molecular. Because funny enough, nano, however, is associated with small. Actually, if you compare a nanomedicine to a traditional medicine, it's much bigger. Because typically molecules used as a drug are either small molecules of tens of carbons atom together, or they are biologicals, molecules such as proteins, antibodies, or small peptides. Again, hundreds of or maybe thousands of carbons unit, if you like to use that. In nanomedicine, we create a much bigger object in that sense. The arrangement of molecule has an extra level of complexity, and we call this supramolecular arrangement. And this ability of creating supramolecules allows to then combine function in the same way as I already described the quantum dots and carbon nanotubes, things that do not exist singularly, but is the collective combination of these molecules that makes them possible. And so now you start to have effect which is associated with multivalencies. So you can bind to multiple receptors, not just one. You can create selectivity based on the composition of the cell that you want to target rather than having a single protein to target, right? And then you can manipulate cells in a completely new way because you can alter the way that cellular cells can transfer materials across so they can internalize materials. So this is a new therapeutic dimension. So they has opened up a lot of opportunity for us to create a complete new class of drugs, which we can then tackle challenges such as Alzheimer disease for creating and proposing new therapy.
How cool is that, Professor? Because you're saying, we getting from the macro world to the micro world, and we entering the world maybe where healthcare will be more precise. We entering a world where maybe it's like precision healthcare through going to probing into this nano world. And you spoke about self-assembly of matter, soft matter, and also atoms. I want to kind of get into that because it's such an interesting topic by itself. But will be great if you could, talk about this breakthrough which happened, where you kind of reversed an Alzheimer's symptom in a mouse using nanoparticles. Will be great if you could talk about that.
Like I was explaining to you, Alzheimer is associated with a lot of things going wrong into the brain, right? One of the major changes that happen during Alzheimer is to do with how materials get in and out of the brain. The brain is very unique. It's very complex. And opposite to any other organ in our body, has its own unique composition. And this composition is highly controlled by our physiologists to the point there is a dedicated vasculature, a dedicated vascular system to the brain, dedicated blood vessels. This is normally known as the blood-brain barriers, collectively known. And this vascular system is being a major challenge in neurology because from a therapeutic point of view, is one of the most impermeable barriers that we have in our body because naturally the brain is defended very, very closely. So it's very difficult to design small molecules or even biological molecules that can cross that barriers and get inside the brain. And therefore, we don't have a way to treat neurological disorder because we don't have a way to get this stuff in there. And so for many years in my group, we've been trying to understand how this transport occurs because on the other side of the story, the brain is also the most expensive organ in our body. It consumes up to 20% of your metabolic cargo For example, any glucose that you acquire during your food intake, 20-30% of it goes to the brain, even though the brain is only 1% in mass of our whole body, right? So there's a huge kind of a dichotomy there, a very highly guarded, extremely impermeable barriers, but at the same time, very expensive from a metabolic point of view. So in other words, why many things cannot get through, other things such as these metabolites, which are important, can get inside very effectively, in very high quantities. And so we try to understand why this dichotomy works, and we identify that the cells that makes the vessels in our brain, the blood-brain barriers, have a very clever way to recognize cargo using how many bonds this cargo can form with the cells. Or, if you like, valency. This is a jargon term to understand how many bonds anything can form to each other. And so this valency control allows this cell to recognize whether the cargo is good for the brain or not. And of course, at the same time, it also recognizes cargo that is good for themselves, right? So they have a kind of a clever way to sort out materials. So we identify these pathways. We discover that if you target this process with the right composition of bonds, then you really have a very quick entry into the brain. And you can get almost anything we want into the brain, and we demonstrated delivery of proteins, nucleic acid. We have a company in the UK now is trying to do gene therapy based on this idea, etc. And this concept is being very well established now. But also, in parallel, we also discover that these pathways is also associated with disease. And in the case of Alzheimer, this pathway becomes less and less functional. In other words, the ability of the blood-brain barrier to transfer material across in both directions, brain to blood, the blood to brain, becomes more and more deficient with the disease progression. And as a consequence of that, you have the accumulation of really toxic waste into the brain. And the most famous of all be amyloid-β. There's another protein called tau, etc. And we know from Alzheimer patients that these accumulation are often associated with disease states. And indeed, many therapies that are being developed currently are very much focused into trying to get rid of these toxic proteins, these amyloid-β, etc. What we discover is that if we now target these pathways, and we know that this pathway is associated with the amyloid-β transport, and then we kind of target in a way to get inside the brain like we did for many different type of delivering efforts, we seems to send a signal to the blood-brain barriers, and that kind of signal kind of reverse that kind of degeneration effect that happened during disease. It's like all of a sudden, we always describe as this pathway is being clogged up by this toxic accumulation, the accumulation of these toxic species, and then our particles, by triggering the right messaging to say, look, I want to get in the brain, I have the right signal to let me through, somehow all of a sudden the cells just wake up again and reopen and recover and restore this transport mechanism. And by doing so, effectively, not only we can get into the brain, but all of a sudden the amyloid-β that is in the brain comes out. And so there is a kind of a kick start of this process, which is a natural process of riddance of toxic waste. And this basically we show in a genetic model of an animal where it's designed to develop this accumulation of toxic protein, amyloid-β in particular, is that basically after one administration only of this supermolecular drug, these molecules, they have more than one function in one. We saw that literally in 30 minutes after, more than 40, 50, up to 60% in some animals of the amyloid-β, which normally is found in the brain, is now in the blood. And so it's kind of open up the, if you like, the tap of removing the toxic species. And then even more surprisingly for us, we seem to have a huge recovery effect across the function of the vasculature, not just this simple mechanism. Many protein goes to the right place, which otherwise would be deficient during the disease. And then the effect on the animal was that 6 months after the treatment, we were seeing a huge improvement of cognitive. Well, not a huge improvement of cognitive. Normally, the diseased animals start to lose cognition and become demented. Effectively, there is a loss of cognitive function, synapses, signaling, etc. But the animal that we treated had no degradation, the degeneration anymore, and they actually treated the treated animal behave exactly the same as the wild type animal without the disease, right? And this is, I don't know whether this qualifies for reversing, but in a way we stopped the degeneration process and effectively stopped the disease. And indeed, that was really surprising is the length of the effect. Only after 3 administrations, when the animal is 6 months old, then we measured the condition right after, and it was already improved. But then at 18 months, 6 months later, the treated animal was still behaving like the healthy animal. So a very, very long-lasting effect. And this, of course, now is telling us, well, first of all, it's giving us a lot of joy because we found a very, very powerful therapy, but also it's telling us that actually the vasculature has a huge implication on the health and the management of the brain function, etc. So it's a really good target, and then becomes an interesting opportunity to develop complete new therapy for Alzheimer. And actually, we discover in other dementia we see exactly the same effect using the same approach.
So, Professor, that sounds really, really, really interesting. So, what you're trying to say over here is that largely amyloid-β accumulation, as well as tau, these are toxic waste which is accumulated in the vasculature of brain, is responsible for Alzheimer's. And your approach, to your nanotechnology approach, where you use supramolecular drugs to deliver via injection into this mice model, which cleared up this toxic waste accumulation, this amyloid-β as well as tau. So that sounds really, really cool. But from a mouse to a human, that obviously, there's going to be like some kind of a challenge, because, from how do you go about what's the next step? From the mouse, do you take this experiment to a primate model like a monkey and then go to a human? So what's this timeline? How do you kind of scale and, bring this from the lab to the market? It will be great if you could talk about that.
This is exactly what we are now doing. First of all, we are, on one side, we try to understand more and more how this process really helps in preventing the neurodegeneration, therefore stopping cognitive decline, right? We are really working beyond the vascular to understand the brain cells, things like astrocyte, microglia cells. They are also involved in maintaining neuron and neural function, right? Second, we also discovered the universality of it. So not only we see this in Alzheimer model, like I say, we saw it in other dementia models, similar effect. And so we're looking at other genetic models, whether they copy, for example, tau transport and alpha-synuclein proteins, which are another protein they are associated with certain type of dementia, etc. And then in terms of clinical translation, so to make sure that this gets translated into humans, yes, unfortunately, there is the second step is to move to an animal model which is closer to humans. However, in those models, you lose that genetic control we have with mice. Getting the same pathology that you see in humans is a little bit more complicated. And those models normally are critical for two reasons. One, toxicology, because of course the physiology is much closer to the human beings, so make sure that there are not negative effects associated with your therapeutic ones. And two is to understand how neurologically this can have an impact, etc. Right? Once you prove that, then effectively you have sufficient data to then apply for what is called a clinical trial applications. This, however, is not just about the testing in animal testing. There is a lot of work, which often we forget, in make sure that the materials are up to scratch. Because if you want to go into human, you understand it's not something that we can produce in the lab every day. This material needs to be produced under certain standards. The quality control needs to be super high. There needs to be really clean manufacturing facility for you to produce sufficient things. So there is a lot of that formulation exercise that really goes alongside. And then normally the way that you do this is through private investors and private ventures rather than public. So right now we have we founded a company in China with my colleagues there in West China, and now we're in the process to set up another company in Barcelona to kind of replicate the same studies. And typically, the way to accelerate this clinical progress is, while Alzheimer's is the most important disease because it's the one that affects more people, you often try to get a pipeline into a more sort of easier onset or subset of the disease where you have a lower number of patients and it's a much faster disease progression and allow you to get to the clinic faster. And so we're going to focus on two particular classes of dementia where it's a bit easier. The energy activation to get to the clinic is a little bit lower so that we can get to human sooner rather than later. And then once we prove that perhaps we can then justify a much larger clinical trial on Alzheimer patients, etc. Right? It's a long way to go and a lot of money. Average talking, we're talking about from now that where we are into something that becomes a drug, we're talking about an average of 12 to 14 years of work and billions of dollars of investment.
I hope that this lab to clinical, transfer, the timeline kind of reduces, because, through COVID, we kind of saw that, what normally, takes from a lab to market timeline takes somewhere around like 8 or 10 or 12 years, can be reduced, if we kind of collaborate and because like you said in the beginning of the conversation, there's somewhere around 57 odd million people suffering at this point in time. And those numbers are only growing. And there's a very, very dire need of intervention, which truly kind of, helps. So, more power to you and your team. And I hope that, there's more investors coming in and there's more collaborations, which kind of accelerates your work, Now, there are other scientists and researchers also, besides working on a mouse model, they also use something called as organoids, where they kind of create, take a single cell of a human and create these brain organoids and kind of experiment. Is an approach like that you're working on? And just wanted to kind of understand, when you say that, these are Alzheimer mouse models, what's the process of, engineering these animal models which faithfully replicate the complexity of Alzheimer's in humans?
Organoids and cellular models or in vitro models, it is an area that everybody, and including us, is very much interested, particularly because they allow you to work with human biology very quickly, because you can take human cells and construct some sort of organoid, organ on the chip in the lab. And that allows you to really understand more the mechanistic, to make sure that what you see is not something unique of mouse, but also is observing humans, etc. So there is that effect. Unfortunately for us, this type of model are yet not there in level of complexity. For certain area of medical development, for example, dermatology, the skin is a rather easy organ to replicate in the lab in vitro. A brain, it's a little bit more complicated than that, because it's not just about putting neurons together and then astrocyte and glial cells, and then we're talking about the vasculature here and the intricate relation between the vasculature and the brain. And that is a very, very dynamic relationship, because the brain continuously alters the blood flow and make sure that there is needs for the neuronal needs. To give you an idea, if you talk to psychology, one of the favorite technique for them to look at the brain function is called fMRNA. fMRNA works on looking and tracking glucose intake into the brain, and that glucose intake is completely regulated by the blood flow. And so these sort of sparky illumination things that happen in the brain, this is really a nice example of the brain control the vasculature very, very intimately. So that kind of dynamic relationship is very difficult to replicate in vitro. And so inevitably you need an animal model, right? And so now, of course, in terms of ethics and practicalities, the mouse is the easiest one to assess. What can we do with that? Of course, the brain of the mouse is considerably less complicated than a human one, but yet there is a lot of conservation and homology between the two. And one thing we can do in the mouse model, we can genetically alter certain traits in order to replicate what we see in humans. For Alzheimer, we know, for example, that why the majority of the cases of Alzheimer are called sporadic cases. They are basically associated with aging. People getting above 65, 60 years old, they start to develop these sort of symptoms. But there is a subclass of Alzheimer that is genetically associated with certain mutations. It also occurs in younger patients, in some cases even 40 plus years old. And these Alzheimer kinds of types are associated with specific mutation of specific proteins. Now, what we know from that, if we can replicate the same mutation into the animal, then effectively you replicate the disease into the animals, and you have almost the same symptomatology that you observe into humans. The brain lose functions, it gets inflamed, you have activation of certain immune cells, you lose vascular function, you start to produce a lot of amyloid-β, a lot of taus, and eventually you lose cognitive and there is a huge cognitive decline. The animals start to be demented, is incapable of doing many functions. And so that is, and like that, there are many examples. You can introduce mutation on the Alzheimer's side. You can introduce mutation on the tau side to replicate the same things with tau. You can do the same with the Parkinson. You can do the same with Huntington, etc. So it's a very powerful tool because it allows you to replicate a little bit more the complexity, right? Yet none of those are humans. So you eventually have to deal with a human testing, and that is the clinical testing eventually. And yes, what you say about COVID was true. Collaborations and then resources really are critical in order to accelerate this timing. But I want to also make sure that it's clear the brain and neurology is a very complex endeavor in terms of clinical translation, because the disease occurs over many years, so you need to have inevitably that kind of timing. While in vaccine, you vaccinate a person, a few weeks later you measure the response of that, and you will know whether that person get infected or not, whether that person has the right immunity is there. In Alzheimer patients is much lower. The clinical trial takes months, if not years, before you start to see meaningful data. So that is inevitable. A practicality limitation that even with resources and collaboration, we cannot accelerate. So I would say a minimum of 6 to 5 years is critical to get the right clinical testing in place, plus the resources. So, I mean, and to give you numbers, right? One of the major problems in medicine is the way that we develop drug is extremely inefficient. And average talking, a drug that gets into the first phase of clinical trial and then makes all the way through is the percentage of those is about less than 10% across diseases. In neurology, that number goes down to 2%, to 2-3%. So it's a very much more difficult endeavor than in other clinical areas.
Right. So, Professor, if there's people who are listening in at this point in time, possibly their family members, who are going through this problem, what are the current interventions which is available at this point in time in terms of drugs? If there's anything which is FDA approved, it will be great if you could talk about that. And who are the other peers who you look up to, who you think are asking the right question, or the right direction, work wise, possibly maybe trying to not just find a treatment but a cure for Alzheimer's?
I mean, actually, Alzheimer for many years it was considered a very difficult endeavor, and inevitably most private company and pharma stay away from it because it was so difficult to get something through, and so it was too high risk for them to try to change. But things have changed recently. There's been an explosion of attempted to try to develop these anti-amyloid-β drugs, and two of them actually made all the way through clinical testing, and they got approved by the FDA a few years ago. Both of them are monoclonal antibodies. It's a type of protein that somehow stick to this amyloid-β and then ask the immune system to remove it, right? It's like similar to what happens during infection. You have an antibody that recognizes the pathogens, and then our immune system remove it. So you do that with amyloid-β. Problem with that are two. One is the quantity of this protein that gets into the brain, because of the blood-brain barrier is very limited. Even though during Alzheimer and late stages, the barrier is now compromised, so there is a little bit more passages, but still not enough. And two, getting the immune system into the brain is not always the right things to do, because you get inflammation, you get some really complicated side effect associated with this immune response in the brain. So there were cases in the clinic that they have to be stopped because the side effects were over. Unfortunately, however, both drugs didn't make it into the market stage because they discovered the actual effect on disease modification weren't huge. Only 25% or 30% of slowing down decline was observed in most patients. And this, of course, is giving a lot of things people to think. And there is a huge pipeline in Alzheimer. There is over 100 drugs being tested right now. Some of those much more promising than others. Yes, there is, of course, a synergy in between what we learned about Alzheimer and the way we develop the drugs. Inevitably, the large majority of these drugs went for, the old view Alzheimer based on cognitive decline, so try to stop that, but that normally becomes palliative. It doesn't reverse the disease. Now, we've seen the same thing with amyloid-β and tau. If we try to target those, yes, we stop some of the cognitive decline, but not necessarily stop the disease. And then what really interests me is the vascular elements. The vascular element not only is a great target because it very much, by maintaining its function, you actually stop not only the accumulation of these toxic waste, but also the immune system really depends a lot on the vasculature to get activated, etc. So there is a lot of feedback mechanism in between the two. But also gives us a much better information how Alzheimer's started in the first place. And so many of my colleagues, including ourselves, start to think that Alzheimer actually is a cardiovascular disease. It's something associated with what is in your blood rather than what is in your brain. And so if it's true that our therapy shows to be so effective even in more complex animal models, perhaps once in humans, and then many of my colleagues, especially in the cardiovascular area and the blood-brain barrier, are showing exactly this huge relationship between the vasculature and the brain, actually it gives a very strong and very quick indication to lifestyle changes, diets, avoiding certain type of foods, etc. There is a lot to study now showing a really huge correlation between the obesity and dementia, of course. And now, for example, the new drug, the anti-obesity new drug, that seems to have a huge effect in slowing down the occurrences of dementia, etc. So I think this is a really important, quick win that you can get out of this type of research. Now, transforming these into drugs, like I explained to you, it takes a long time, and it will take a long time. And so for the patients currently affected, they might be a bit too late by the time we get there. But at the same time, try to understand we are really making huge progress in understanding the brain more collectively. It's not just longer neurology, where you're looking at neurons, which is only one of hundreds of different cell type in our brain, right? For each neurons in our brain, there is 10 astrocytes. There are 8 microglia cells, which are the immune cells of the brain. There is 12 endothelial cells, which make the blood vessel. There is 5 pericytes, etc., etc. It's many more are cells that for many years we've been ignoring them because we thought neurons are the most important one. Now we're looking at them collectively. And so this collective approach is really making a huge impact in how we're looking at the brain function, disease, and of course, developing therapies in that sense. So I'm confident that we're going to have a lot of more things coming up in the next years.
Yes, Professor. So exciting things happening around the world. So I hope that this multi-directional ways of asking a question more holistically, I think will accelerate, maybe us finding a treatment or a cure of Alzheimer's, because like we said in the beginning of the conversation, 57 odd million people in the world suffering from Alzheimer's, and those numbers are only going to grow. So yes, there are, people looking at reversing this and through the lens of brain-computer interfaces. They're looking at treating this through ultrasound. They're looking at treating it through the lens of genetics, and then you're looking at nanotechnology. And recently I read this news about a team from China, from the Academy of Sciences, Kunming Institute of Zoology. That's very, very interesting. They are using sound, which is completely non-invasive. And they did this for 9 weeks, is something which I think I read. They were playing the sound, a 40 hertz frequency, to an animal model, a monkey. And the researchers kind of discovered after playing that sound for almost like a hour each day, that the levels of amyloid-β in the cerebrospinal fluid had increased by 200%, which indicated that a considerable amount of plaque had been removed, this toxic waste that we talking about. So what are your thoughts on something like that, an approach which is completely non-invasive, using just sound, which is 40 hertz frequency, and which is helping in removal of this toxic waste? That's my first question. And who are the other peers, according to you, who are asking the right questions of what Alzheimer's is and how we can possibly find an intervention or a treatment and a cure for Alzheimer's?
Well, I mean, in that particular study, they only showed a reductions of A beta and a removal of A beta. In that, I don't think the data show any effect on cognitive decline, etc. So we don't know whether the real effect is then transformed from that. I mean, the brain is based on a combination of neural signaling, etc. And that signaling is not just something that the brain gives us to our body, but it's also something the brain acquires from outside. And of course, sounds, visions are input in our brain. And these are converted into certain neuronal signaling that then transform the functionality. Like I explained to you, when you do fMRNA, if you stimulate, let's say, a part of your memory, this part of the brain light up. If I stimulate the vision part, this other part of the brain light up, etc. So this light up means there is a certain functioning, a certain combination of elements, neural stimulation, vascular stimulation, etc. So I'm not surprised that some of these have this kind of effect, right? They can be positive in the sense, in this particular case, this particular frequency showed to have a huge reduction in the A beta accumulation into the brain. It's fantastic. That's, I think, a great way to show that actually manipulating our body is not something that we only need molecules to do that. You can use physical stimulus for doing that, in this case, sounds, right? But ultrasound is also being used for favoring and getting stuff into the brain. There is some really nice study on Parkinson people where you introduce electrodes and electrostimulation in a particular part of the brain, and that really helps a lot. So this idea of therapy is becoming so multidisciplinary. Yes, they really are embracing more and more discipline into that. So I'm very glad, because we learn a lot. And then you said to combine. What if we combine our drug with sound stimulation? Will it improve things? And again, in biology, it's not a simple additive process. Not that 1+1 is always 2. 1+1 might become even more than 2, because there are sort of phenomena that characterize life in general, which is called emergence. Is this fact that by putting things together, you create a new type of energy that come from the collectiveness of things rather than the way it is. And this is, I think, the new mindset that we are now in science trying to embrace as much as possible.
Right, yeah, exciting times, this convergence of sorts, and there's so much we don't know, but we discover collectively through these different approaches. Who would have thought that just basic sound frequency, a 40 hertz sound frequencies, can play a big role in Alzheimer's. And there's so many exciting things happening in the space of neurotech and neuroscience. There's companies such as Neuralink, Pandromics, Synchron, and others, and trying to understand the language of the brain. I mean, 80 billion neurons and 100 trillion synapses. What are they kind of doing? And is there a language going on in the brain? And if we kind of understand the language of the brain, I mean, structurally and functionally, if we kind of understand the brain, how cool it will be, because there's millions of people in the world who are suffering from neurological disorders, and what if we could kind of reverse that? So through this approach of yours, through these nanoscience, nanoparticles that you're using, I mean, beyond Alzheimer's, are you also looking at tackling maybe other neurological disorders?
Absolutely. I kind of mentioned already that we are now exploring other type of dementia, neurodegeneration. We're just starting looking at other diseases. Another really, really horrible disease affecting the brain is called multiple sclerosis, which is a chronic inflammation of the brain that starts to degenerate the brain. And again, we see how our approach of tackling the blood-brain barriers in a certain way really has a huge impact in how this disease can be stopped and managed. We have done work in the past in looking at glioma, which is a brain tumors. Again, in the case of brain tumor, is an interesting fact that once the tumor develops, there is a part of the tumor that stays where it was born and become like any other tumor, quite misfunctional, etc. And this is, although it's still in the brain, it's relatively easy to treat, but actually often the damage comes from those cells that escape this core, the tumors, and actually what the brain tumor cells do, they often follow the vasculature as a way of scaffolding the movement and then exploit at the same time the high nutrient that these cells give them. And so understanding this symbiotic relationship between the tumor cells and the vasculature becomes critical in managing brain tumors, right? So we done work in that area too. Then there is a instead of using this sort of new approach to supermolecular traditional, we also done some genetic therapy where we deliver the gene in order to treat a very rare genetic disorder, etc. So there is, the brain is being a part of the body that we've been studying for a long time, and indeed has taken a lot of our interest in doing things.
In the course of the conversation, you did speak about self-assembly, but I think self-assembly is so awesome. It's something which really excites me. Because our world is quantum mechanical, and we are made out of atoms, and how these atoms kind of clump together. With the four fundamental forces: the weak and strong nuclear energy force, electromagnetism, and gravity. When they all kind of combine and come together, the magic happens. The magic of creation. Everything kind of comes into being. The earth, the moon, stars, the air, the trees, you, me, and everything. So the self-assembly concept is so very interesting. So it'll be really cool if you could kind of explain what self-assembly is to my audience, and how does these atoms kind of come together, from soft matter of the atoms, how do they come together and like a Lego block and kind of, build everything, and what excites you most about its near-term application, and what futuristic possibilities do you, envision?
Sure, and this is an area which is, very quickly become very philosophical, because one of the areas that we've been, some time entertaining with is how our tools of making materials by copying biology actually becomes a way to understand biology. In a way, if you think about how an organism evolved, they become this sort of broth of chemicals. All of a sudden, they become a single cell capable movement, reproductions, metabolism, etc. And at the end of the day, how this assembly of molecules then becomes alive is a very fundamental question in science, and still no answer, right? There are many different theories. There is a theory of the Frankenstein theory from Stanley Miller, where this broth of amino acids all of a sudden start to come together, or there is the mRNA world, the submarine vent world, etc. There's many things. All of them agree on self-assembly. You need to put molecules together, but this molecule together needs to come in a certain way, because that certain ways give you this extra energy that then drive more and more complex transformations, and then these complex transformations again come together, and this becomes more and more. So there is a kind of a cascade effect of energy based on information that you generate that start to then becomes alive, right? I love a sentence which I always paraphrase from a very famous physicist called Ilya Prigogine. He was a Belgian-Russian physicist based in Brussels, which was very famous for a branch of physics called out-of-equilibrium physics, right? He's tried to understand how things, and he always said atoms and molecules are immortal, but as soon as they come together and form more complex structure, they become mortal. And that's exactly what it is life. If you think about it, as we are made of certain atoms like carbon. If you think about carbon, coal is there forever, right? If you think about nitrogen, is in the atmosphere, oxygen, water, right? What is the expiry date of a water molecule? There is no expiry date, but you put water together with the rest and you form a single organism, self-organism that start to have a birth and a death. And this transformation of this molecule together in life is becoming fantastic. And all of this, at the center of it is not just about the chemistry nature, is how these molecules come together, how they self-assemble into very specific structures. And nature is very good at that. It's beautiful in this arrangement. Of course, so beautiful that we have to invent a new type of mathematics in order to understand those patterns. And again, this is really what really feeds most of our research. In really understanding not the chemistry, not the physics, but the combination of the two. How this putting together creates sort of new emergent behavior, and now this emergent behavior can be used for more new functions, etc. Right? Of course, nature has done this in over billions of years. We really had the beginning of this path, but I think it's the right way to look at things collectively, and not looking at the component, but more about what the components becomes once together, right? And this is a complete new approach in that. But self-assembling is can be simple, right? One of the most simple forms of self-assembly is soap, right? You must have played when you were a kid trying to make soap bubble. That is a great example where you take water that normally doesn't like air, and then you add the molecule called detergent, which has the ability to having one part that likes water, one part likes the air, and then you create this sort of beautiful bubble where the membrane that closes it up is formed by these molecules all line up together in a specific way, and they create this sort of new mechanics. Even though the molecule locally are liquid, actually be almost like a solid things, right? And all of these beautiful example of self-assembling. And then, the same principle dictate how cell membranes forms, protein, DNA, etc. So, and of course, if we can replicate it synthetically, it's a complete new tools, not just to manipulate biology and creating new drug, but also really understanding how biological organization comes about.
Eric Drexler, talks about nano assemblers, and Ray Kurzweil also has spoken about nano assemblers a lot. What are your thoughts about nano assembler? Do you think we could ever be able to kind of build a nano assembler? And will be great if you could also maybe explain to our audience what a nano assembler is. And Eric Drexler also talks about a world, maybe futuristic in the future, possibly, maybe we kind of tinker around, with nanotech, and eventually maybe we could, build Gray Goos, which could kind of, completely destroy our world. It also will be great if you could, talk about nano assemblers. You think we could build them? What is nano assembler, and your thoughts on Gray Goo?
Well, I mean, Dexter and nanoassembler are still not a reality, and we are really far away from that kind of level of complexity or this kind of intelligent behavior of molecules. We are exploiting certain natural process driven by supramolecular forces in order to create structure ever more complex, but making this then structure intelligently, collectively organized within itself, and we're really far away from that. We have now more elements, and I have a colleague here in my institute that is very good at that, of creating what we call nanorobotics, where you start to mimicking movement. We work in the past in introducing movement to our system, which is again chemically driven. So you do what is called chemotaxis, a movement that is driven by certain chemical gradient, right? And again, this process can be possible. From there to really then have a remote control nanoparticle that you can tell to things, this is sci-fi. It's definitely not yet, not even close. I wish, but on the other hand, nature does that. Nature has this ability of self-healing. They reproduce and creating complete new life where there are examples in nature where a single cell organism become a multicellular organism that become more complex, etc. Right? Our body is a great example. The brain is a fantastic example how you assemble neurons in a certain way. You create a very unique computational power machine that is our brain. So I think where we are engineering-wise, we are really far from Drexler vision and nanotechnology. I think it's a nice horizon set challenge, and I like that. This kind of sci-fi often becomes inspiration. Philosophy becomes inspiration. I think you need to have that kind of long blue sky view. Is critical, but we're not there yet into have these sort of nano machines that can one day cure our disease. Things that you see in sci-fi movies now more and more often. I don't think we're there yet, and there is a lot of work to do to get there, because that means really getting to the level of control that biology has. To give you an idea, a single cell organism, in order to be functional and be considered alive, still need 600 genes together. 600 genes means this is times at least 2,000 in terms of proteins. And then on top of it, you need all the lipids, the sugars, the fat, all together in a certain form. So the level of complexity to become alive, to the level of cognition, is still very far from what we can achieve. We know the level of complexity is still far from our reach. But we're understanding more and more. And I think one day perhaps we're going to get there. But as usual in science, things can be used for good and bad. We scientists always go for the good. And hopefully we leave it to the judgment of people and users to use them and always for the good. But unfortunately, sometimes things get a little bit out of control. But the Gray Goo and the book written about it is a nice story, but no more than that. There is no scientific evidence we're going to have a Gray Goo anytime soon, I'm afraid.
Right. Right, right. Professor, I mean, there is artificial intelligence, it's kind of, completely taken over the world, and it's like, I mean, we are at the cusp of, what people kind of say that, maybe in the next possibly decade or so, we should be able to kind of, build maybe general purpose machines, machines which could possibly think like humans, and there is a convergence of sorts with healthcare, and largely, here we are with, when it's healthcare, we're dealing with data. So, what's the role of nanotechnology and AI, and how do you think AI is going to help accelerate, your experiments as well as your lab to market timelines?
Sure. I mean, nanotechnology had a huge impact in AI because has had a huge impact in electronic industry and creating new, more powerful chip and transistor, etc. And this is what behind then the GPU, the CPU that drives all the AI that we need, right? So, and this is probably going to get better. There are things like quantum computer where nanotechnology become critical in order to construct these devices, etc. So there is a lot on the hardware side. And then feedback. AI is a tool nowadays, is becoming ever more used. It's a very powerful tool to do science. Because allow us to do calculation in a completely new way, right? Not necessarily you reproduce human behavior, etc. But one thing that people forget about AI, the AI has the ability to navigate multidimensional space like no human can do. In a way, typically when you look at the scientific graph, you always have a one axis and the other axis, and you look what happened. So you have two factors that you look at. AI can do this multiply times n. And you can look at things and analyze data now in a space that allows you to discover more effectively patterns. And so we also in the lab are now implementing AI as we speak because allow us to really create a much better design tools. It allows us to understand our data more often, better, and identify patterns, which is like I explained, especially in biology, nothing is linear. It's not that 1 + 1 is 2. It's not that because I have more, I get better. Actually, sometimes you have a behavior which are what is called non-monotonic. So all of a sudden you have a response and then disappears. And so understanding these sort of really unique windows where everything works or doesn't work is critical. And that means experimentally you will have to go through billions of experiments. And now using this new tool of data science, you can reduce that space considerably because now your analytical power is much more it's much more capable of dealing with this multidimensional space. And then, of course, you keep feeding data into these networks, and then again, you get more data out, etc. So this is inevitable. We need to embrace AI as a critical element in our work. Not just about writing and reading better, but it really is the way we do science that makes a huge difference. Whether one day AI will become more clever than we are as a scientist, I don't think we're there yet. It definitely, and I think humans are very good at adapting, much better than AI. So I have a feeling that as AI becomes better, we're going to become better, right? Yes, it's true that certain things will be lost. Like I don't know, I cannot memorize any phone number anymore because my mobile does it for me. It doesn't mean I'm more stupid than before. It means that now I can have a different type of information in my brain that I have to use for memorizing mobile numbers now. I don't need that. So I want to believe in the same analogy with AI, as AI will give us access to a completely new type of functions, then our brain can adapt to that. Or after all, AI is templated on the human brain. So I want to believe that our human brain is still better than that. And so we are able, of course, not every humans will do the same. There will be jobs that will be completely replaced by robots, I guess. It's happening already. For example, in coding, in informatics, in IT, many old coders are not needed anymore because AI can do this better. But again, I think it's very important we adapt and embrace these changes as quick as possible. Definitely, nanotechnology AI has had a huge impact. It accelerates our discovery process considerably.
Beautifully put, Professor. Can you share any updates from your molecular bionics lab and your startups, Bionautics?
Yes, well, we are in the process of getting more and more data on understanding the brain. In this transport process, like I explained to you, we've discovered that the role of the vascular is beyond Alzheimer, is actually associated in many other diseases, and it becomes a very fantastic opportunity for us to create a complete therapy across the world, across different type of disease area, not just dementia and Alzheimer, but also this idea of making drug in a completely different way, right? So this idea of creating sort of emergent drug. It's a drug that really combine things together and looking at things in a different way, forcing the traditional medicinal chemistry, which is always based on medicine, on biology and chemistry, to also embrace physics and mathematics in order to really look at. So this kind of a philosophical push into extend the multidisciplinary to discipline that normally you wouldn't consider, but now that you are now embracing more and more on that. And AI is really helping in that as an example. And I think this is where we are now going there. We start to look at new models to predicting drug behavior in the body, almost sort of similar to what you do with the forecasting weather and looking at behavior more sort of a more collective way. And again, thanks to the new computational science advances, we can now, getting there. And I think this is going to be a huge impact in how we design drugs eventually. All of this, however, kind of suggests a completely new way of looking at therapy and making it even more personalized. Because my biology, being human, is the same as my brother biology, which we share a lot of DNA, but not necessarily we are the same. And inevitably, not necessarily we respond to the same chemical in the same way. So therefore, the drug will work differently. And this, I think, is a factor that we often forget in medicine, but this necessity of personalized treatment becomes more and more. And this is not just about making the drug more precise, but also understanding how point of care therapy can be adapted. Perhaps pushing the manufacturing of the drug there at the point of care in the hospital and say, look, this is your profile. We need this drug. And I program a 3D printer that makes that drug there and then, right? So this is, I think, at those sci-fi and at this stage, I think this is one of the challenges we need to start to adapt. And nanotechnology is perfect for that in that sense, because the concept of self-assembling is very easy to translate in this type of context.
Right. Professor, my last question to you. What's your personal moonshot? And it'll be great if you could paint a picture of the world. What's the future of healthcare according to you? I mean, maybe in the next 5 to 10 years, what's the future of healthcare going to look like?
Well, the future of care that I want to see in the next 10 years is definitely a future where Alzheimer becomes a monomodular disease, which is not, where we start to understand more and more our brain, so therefore we can predict diseases more and more effectively. And definitely we have this level of personalization down to the single patient and really exploiting the large amount of data we're collecting clinically from patients to transform this into meaningful medicine, right? Really this feedback mechanism of personalization and precision medicine that is critical. I think this is what we need. And this is what I think is going to happen, is happening already. And I think inevitably as a drug makers, we need to adapt to this process. I don't think we're there yet. Our drug making is still a little bit cumbersome. So I think, however, nanotechnology is pushing into that direction. I think in 10 years' time, we're going to have more and more these sort of nano drugs and nanomedicine drugs happening. I don't think we're going to get the nano assembler yet, but maybe in younger years, that's the new future, getting those Dexter nano assembler that really cure disease in that way we see in sci-fi. That would be possible once we understand this collectiveness and make them really clever, right? We're not there yet.
Right. Professor, really, really appreciate you taking time and being part of the podcast. I think we're sitting in a fantastic point of time. I think globally, we are trying to understand the brain, because if we understand the brain better and if we are able to maybe reverse engineer the brain, there are so many downstream applications. We will be able to possibly build machines which are maybe as intelligent as us, because right now I think the current paradigm of building a machine that's intelligent like us is definitely failing, because we're throwing in compute, we're throwing in data, and we hitting the scaling law. So if we are able to reverse engineer the brain, we will be possibly, we will be able to build a machine which possibly is intelligent like us. And also maybe once we understand the brain, maybe we'll be able to peek and probe and find answers, and possibly not just look at treatment, but also cures of these neurological disorders. So please continue doing what you're doing. Wish you the very best. And to my listeners, if you like what you see in here, then please press the subscribe button. Until next time, see you guys. Bye-bye. Thank you, Professor. Really appreciate this. Yeah. Thank you.
Thank you very much.